Good evening to aspirants. So there was some technical glitch. So now hope we are live. And what we'll do today is that we'll try to understand biotechnology, some portions of biotechnology which are exam important for this prelims. I hope I'm audible. Just a second. Technology, some portions of biotechnology which are exam important for this prelims. Okay, so I'm live. So what we'll be trying to do in this session is to understand biotechnology. So we'll try to develop a basic understanding of biotechnology. In order to understand biotechnology, we need to understand from the very basic itself, that is from what is a cell. So inside a cell, we need to understand some of the important structures. If we are clear with these structures, then we can understand. So all the tools of gene editing that we see. So we keep hearing tools of gene editing like CRISPR-Cas9. So this CRISPR-Cas9. So what are we doing in CRISPR-Cas9? So we are just manipulating the different portions of our DNA. So we need to have a basic understanding of what is DNA. So in this slide that you can see, so if you look at the nucleus, so this is the cell, and inside there is this nucleus. So what you're seeing is this nucleus. So if you open that nucleus up and if you look closer inside, then you will see that there are chromosomes inside. Inside the chromosomes, if you open that chromosome up, so can you see this? There is a thread-like structure. So these thread-like structures are wound on these something called as histone protein. So these tube-like structures are there, which are used for binding this DNA. But if you further open up the DNA, and look deeper, and what you will see, you will start seeing these strands. So these strands that you are seeing, so this is one strand and this is the other strand. So these strands are made up of sugar and phosphate. So you can see alternate layers of these sugar and phosphate. So there is sugar, there is phosphate, so there is sugar, and then there is phosphate. And what you are seeing here, these things that you are seeing here, these are called as nitrogen bases. So these nitrogen bases that you can see, that is guanine, cytosine, adenine, thymine. So on one side you have a particular nitrogen base. So on the other strand also you will have a corresponding nucleotide or nitrogen base, but there is what is called as base pairing rule. So in base pairing rule, you will see that A pairs only with T and C pairs only with G. So here, let us say if this is T on this side, so here you will see that this is A, and they are bound together by some hydrogen bonds. So this one complete set is called as your base pair. This is your one base pair. And if you see, or if you open up the entire human genes, then you will see that there are more than 3 billion base pairs, so in human genome. So the terms like this, what is a DNA? So this part that you are seeing, this part is your DNA. This part you are seeing, this is DNA only. And there are some parts of the DNA which are called as the functional part. So this part from here to here, let us say it is coding for something. It is coding for, let us say here, eye color, or it could be coding for, let us say, a blood group like the ABO blood group. So these functional part in the DNA, they are called as genes. Genes are your functional part in your chromosomes. So functional DNA is called as your genes. So in, if you open up this string, so you will see there are many sorry for that. So you will see that there are many portions in the DNA which are not coding for anything. So those portions are called as what is known as junk DNA. Junk DNA. So there are some portions of your DNA which are called as junk DNA. See, this junk DNA, tell even on this there are debates that can anything be there inside our body which is junk. So functionalists believe that we have not discovered the function of this junk DNA, but they are having some purpose which we will discover sometime later. But as of now, what we understand is that genes are the functional sections. So you will see, if this is your DNA, so you will see some portions which are functional, and you will see some portions which are non-functional. So there are non-functional portions, and there are functional portions. So the functional portion, we are referring it as genes. And you will see there are nitrogen bases. So nitrogen bases are present. So if this is A, T, C, G on this side, so on this side you will have T, C, G, A. So this is the general base pairing rule. If we understand this, then we can understand that in gene editing, what we are trying to do is to make a cut at some places, to join it at some places, and then we can carry, or we can have a desired gene at a particular point. So these, how do we do it? So for that we need to understand what are the gene editing tools. But in order to understand that, we need to understand some of the So in order to understand gene editing, I'll just open the slide. So in biotechnology, we need to understand that what are the tools in biotechnology. How do we go about cutting these these genes or DNA? So for doing that, so we need some enzymes. So if we have to cut a DNA, we need some enzymes. If we have to join some part of the DNA, then we need some enzymes. So where do we get these enzymes from? So we get these enzymes from nature. So the first enzyme that we need to know in biotechnology, or in biotechnology, what we are doing is that we are playing with the genes, and while we are doing that, we need some tools at our disposal. So we have certain tools at our disposal. So we need something to cut the genes, we need something to join the genes, and we also need something to multiply those genes or segments of the DNA. So for that purpose, we have enzymes. So what is the role of these enzymes? We'll see. But before that, we need to understand that why are we able to transfer some genes from some organisms to another organism. So we also need to understand principle of biotechnology. I'll just come back to the slide. So what is this principle of biotechnology? So in this principle of biotechnology, what we are saying that, let us take this example of BT cotton. So when you say BT cotton, in BT cotton, we are saying that we have extracted some genes from a bacteria. So this bacteria is Bacillus thuringiensis. So this bacteria is Bacillus thuringiensis. So this is a common soil-borne bacteria. So we are extracting some genes from this bacteria and we are putting it in cotton plant. So why is it possible that we are able to extract something from bacteria and we are able to insert it in plant tissues? So it is possible because if you look at the DNA, or if you look at the genetic structure of a bacteria, and if you look at the genetic structure of plants, then the genetic structures are same. It means bacteria is also using these ATCGs. So the ATCGs, the nitrogen base pairs that you saw, so bacteria is also using ATCGs. Cotton also is using ATCGs to code. So genes are what? They are coding of information for production of any particular kind of proteins. So cotton plant is also using this, bacteria is also using this, humans are also using this. All the living organisms, what you're seeing, that they are using these base pairs only to code information, to pass on traits from one generation to another. They are using this method only. So that is why even a bacteria, why is a bacteria able to infect us? The bacteria is able to infect us because their genetic component is very similar to our genetic component. That is why they are able to infect us. So that is why we can carry something, we can transfer some genes from bacteria to cotton plant, and we can transfer something from, let us say, human also to bacteria. So how do we make insulin in the lab? So we make insulin in the lab by extracting human insulin gene and we transfer it to bacteria. Now that gene expresses itself inside bacteria and the bacteria itself starts to make insulin. That insulin we extract and we give it to patients. So it means that genes from living organisms, from different living organisms, we can move it, or we can cut paste them from one organism to another organism. Why this is possible? It is because they all the living organisms, they are biologically and chemically similar. Why they are similar? It is because it is said that we all have evolved from common ancestor, and that common ancestor is said to be what? The bacteria. So it is said that, or there is one theory that says that the first living organisms that emerged on this earth were bacteria, and from bacteria everything else has evolved. So because these bacteria were using this DNA method or these ATCGs, nitrogen bases, to code for information, to pass this information from one generation to another, so all the living organisms that have evolved are using this method only. So that is why it is possible that we can transfer genes from one organism to another. Based on this very basic understanding, you can see that in prelims you will be able to answer many questions. We'll see the questions also in the end, but this is what is known as the principle of biotechnology. So the principle of biotechnology says that genes of all living organisms are similar biologically and chemically, so we can transfer genes from one organism to another organism. It doesn't matter that whether it is a lower organism or whether it is higher organism. We can transfer from bacteria to human, we can transfer from human to bacteria. So that is essentially the principle of biotechnology. Now since you know the principle, now we need to have an understanding of the tools of biotechnology. So what are the tools of biotechnology? So what are the tools? What we mean by the tools of biotechnology? It means the tools through which we can do gene editing or gene manipulation, or how do we produce these GMOs, that is genetically modified organism. How do we transfer this gene from, let us say, this bacteria to cotton plant? How do we transfer genes from humans to, let us say, a bacteria? So that answers the question how do we transfer it. So the tools, through use of tools, we are able to transfer these genes from, let us say, a bacteria to a cotton plant. So what are the different tools? So just like in common stationery, we need what? We need scissor, we need gum, we need some Xerox machines. So similarly in biology also, in biotechnology also, we need these tools. So what are these tools? So these tools are known as molecular scissor. The first tool that we need to know is the molecular scissor. So molecular scissors means how we can use or how we can cut DNA. how we can cut DNA. So we use molecular scissors. So what are these molecular scissors? These molecular scissors are some enzymes. These enzymes we call it as restriction enzyme. So we need a restriction enzyme. We need what is known as something to stick the DNA. So we need what are known as ligation enzymes. So we need DNA ligase. So this enzyme helps in sticking the DNA, and then we also need multiplication of this. So we'll use polymerase. So we'll use polymerase to multiply. So this one is for cutting. This is for cutting DNA. This enzyme is for cutting. This is for joining DNA, and this one is for multiplying DNA. So from where do we get these enzymes? So in biotechnology we need enzymes. So these enzymes, you'll see that we get it from nature. So this first enzyme we get it from this first enzyme, the first enzyme, that is the restriction enzyme. So where do we get this restriction enzyme from? So this restriction enzyme, so what you are seeing here in this slide is that you are seeing that there is a bacteria here. So this whole cylindrical structure that you are seeing, this is a bacteria. So this is a bacteria. This is a bacteria. And what you are seeing, that this is a virus. This is a famous virus which is known as bacteriophage. This is a famous virus which is known as bacteriophage. Bacteriophage meaning what they eat. Phage means to eat. So they eat bacteria. So these viruses, they eat bacteria. So what is this virus trying to do? This virus, they are having some genetic material, that is this could be your DNA or it could be RNA. It could be DNA or it could be RNA. So they are sending down this DNA inside the body of the bacteria. So what will this DNA or this genetic material will try to do? So this genetic material will try to multiply inside the body of the bacteria. So what is this bacteria doing then? So the bacteria also develops some defense mechanism. So the bacteria develops this restriction enzymes. So what you can see here, so these are your restriction enzymes. These are restriction enzymes. What are these restriction enzymes doing? So here you can see that these restriction enzymes are cutting this genetic material into small fragments. Because it is cutting into small fragments, the information or the coded information that is stored in this genetic material is lost, and because that information is lost, it cannot multiply now inside the bacteria. So these restriction enzymes are your defense mechanisms of bacteria. So we extract these restriction enzymes from bacteria. So initially during 1980s, not many bacteria were known, so the number of restriction enzymes were very limited. But now we have many restriction enzymes. So these restriction enzymes are called as your biochemical scissors. So they are acting like a biochemical scissor. So these tools we can utilize in the lab also to make cuts of DNA at specific points. So next enzyme that we'll try to understand is the DNA ligase. So DNA ligase are responsible for joining the gaps that form in a DNA during replication and DNA repair. So even in our body, so when our body is injured, or when the cells, they are trying to repair themselves or they are trying to multiply, so they need this mechanism of joining the DNA. So this DNA ligase helps in joining the DNA. So it joins the sticky ends of the DNA pieces from different sources that have been cut by the same restriction enzyme. So once you have cut the DNA, you need to join the DNA. So how do we do that? We do it by DNA ligase. So then the third enzyme is your DNA polymerase. So these DNA polymerase, you can see that they help to replicate DNA and to help in repair and maintenance. Because they are helping in replication of DNA, we are using this polymerase enzyme to multiply the DNA. So these are the enzymes, and we also need what, let us say, we also need sometimes carriers. We need some carriers which are known as vectors. These vectors are what their role is to transfer the desired genes from one organism, let us say from BT to your cotton. So we could be using some vector to transfer the desired genes from BT to cotton. And we also need host. So in this case, let us say this is the host. So how do we transfer the desired genes from, let us say, a bacteria to a host? So there are some methods that we'll see. So when we combine all these tools, so we can go for gene editing. So one of the most famous method of gene editing is what is known as the recombinant DNA technology. So recombinant, or this is also known as rDNA technology. Recombinant DNA technology, or this is also known as rDNA. What does this term mean here? This recombinant, this term, it means combining of genes from different sources. Combining of genes from different sources. So in this particular slide, we'll try to utilize all those tools and we'll try to see how do we go for gene editing. So let us say we have taken one bacteria, a famous bacteria, that is E. coli, and we have extracted one part of the bacteria, that is the plasmid. These plasmids are called as circular DNAs. Plasmids are your circular DNA. In this case, this plasmid is acting like a vector. So we what do we want? Let us say this is the human insulin gene. So this is the human insulin gene. We want this gene inside this bacteria. So what are we doing? That instead of sending it directly to the bacteria, we are using a vector here. So we are using the plasmid, that is a part of the bacteria. So we are extracting this plasmid and we are making a cut. So how have we made this cut? So using RE only. So using RE, we have made a cut and we have extracted this desired gene again by using what? RE only. So we have extracted the desired gene. So this is our desired gene and we have extracted this from a human cell. And then what we are doing is that here you can see that we are trying to bring them close, or we have to stick them. Next, what we are doing, we are adding some DNA ligase. So we are adding some DNA ligase. So now you can see that the ends are completely joined. So this piece, what you have got, this particular piece is a recombinant DNA plasmid. So here we have combined genes from two different sources. So we have combined genes of a bacteria and here we have combined genes of human. And this particular plasmid, now we are inserting it back to the bacteria. So how do we insert this into the bacteria? So there are different methods. So generally what we use is thermal shock. So in thermal shock, what we do is that we'll keep this plasmid, this recombinant DNA plasmid, near the bacteria. So what will happen near the bacteria at quite low temperatures, that is, we'll keep them in ice together. Also they will incubate, so they will lie side by side. And what we'll do, that will suddenly increase the temperature. When we suddenly increase the temperature, then that acts as a thermal shock. So when we increase the temperature, so there will be some pores formed on the body of this bacteria, and now these plasmid can enter through this. So because of the pore, the plasmid enters here, and then when you remove that thermal shock, or when you keep it back again into ice, then it is, this part starts to heal. So this part heals and now the plasmid is inside the bacteria, or let us say this, you can call it as host. So we wanted to transfer this desired human gene into this host, so we have been able to do that using vector and by giving some thermal shock. We can give some electrical shock also, we can give some chemical shocks also. There are other methods also, so that we can use to transfer these genes. And once this gene is transferred, so this recombinant DNA is transferred here, now the bacteria will try to multiply. So bacteria multiplies, and inside every bacteria you are seeing that you have the desired gene. So now these genes will try to express itself. So when it expresses itself, it would do the function which you wanted. So let us say if this was your human insulin gene. So this bacteria and these bacteria, they will start production of insulin, because the insulin gene will start to express itself and they will start to make insulin. So they will start to make insulin, then we'll collect that insulin, we'll purify that insulin, and then it can be given to patients. So this is the general method that we use in case of gene editing. So all these steps that you are seeing, it is what we are trying to produce some genetically modified organism, or in this case what we are trying to do is we are trying to transfer genes from one organism to another. So this is also known as transgenic. This is also known as genetic engineering. Genetic engineering, this is also known as gene editing. So these are some of the words or terms used synonymously. So here what you are seeing, we are transferring genes from one organism to another, we are playing with the genes, so that is why we are calling it as gene editing or genetic engineering also. So in this way we are able to do gene editing. So let us say, what is this CRISPR-Cas9 then? So this CRISPR-Cas9 has become very popular nowadays. So what is this CRISPR-Cas9? Okay, coming with respect to the vectors that you have seen. So in this case, that in the example that you saw, that we use plasmid as a vector, we can use a virus also as a vector, we can use bacteria also as a vector, we can use yeast also as a vector. So you can see that most of the microorganisms we can use as vectors. So there is no restriction that only viruses can only be used as a vector. The whole bacteria can be used as a vector, viruses can be used as a vector, fungi can be used as a vector. And there are other methods so through which we can transfer genes without using vector also. So what are those methods? So those methods are called as electroporation. So electroporation is what we are doing, that we are giving some electrical shock, and because of that the cells develop pores, and we have kept the desired genes in the vicinity. So through that pore the desired genes enter, and then when we remove the electric shock, so the cells or the cell walls and the cell membrane, they start to heal, and then the desired gene is inside. In chemically mediated transfer, it is a similar method only. It is just that instead of giving electrical shock, we are giving some chemical shock. So in that we sometimes use chemicals like calcium phosphate. In presence of calcium phosphate, we see that these cells develop pores, so then the desired genes can enter inside these cells, inside the host, and when you remove it from those chemicals, then it automatically heals. In microinjection, what we do, so that let us say if this is a cell and if this is the nucleus, you want to transfer some desired genes, so we are literally using some injections in this case. So using injections, we inject the desired genes. So let us say this is the desired gene, so we will take micro injection, and we'll inject the desired gene inside the nucleus of this particular host. So in this way we can transfer the desired gene. In gene gun, in some scenarios, what happens that the cell becomes sticky to, let us say, gold particles. So in some conditions cell becomes sticky to gold particles. So in the cell, or sorry, in the gold particle, what we do, we put the coating, or we use the plasmids, and the plasmid is having the desired gene. So inside we'll be having the desired genes. So these are the desired genes, and then these gold particles are fired into the cell, and you will see that the desired gene is now inside the cell. So in this way also we can transfer the genes. So what is this CRISPR-Cas9? So if you look at the full form of this CRISPR-Cas9, clustered regularly interspaced short palindromic repeats. So what is this palindromic repeat? So the restriction enzyme that in this CRISPR-Cas9. So CRISPR-Cas9 is one of the popular gene editing tools, and in this the bacterias, we are using the restriction enzymes produced by bacterias, and bacterias that you saw that they are producing restriction enzymes. Those restriction enzymes have unique quality of understanding palindromic repeats. What is a palindrome? So palindrome, let us say it reads same from both the left hand side and the right hand side. So if you take words like this, little, or so if you read from this side, it reads the same. If you read from this side, it reads the same. So in your genes, or in your base pairs, if you look at this example, if you read from this side and if you read from this side, it is a palindrome. So these restriction enzymes, they identify where the cut has to be made using these palindromic repeats only. So the Cas9 here, the Cas9 term here that you are seeing, is the name of the restriction enzyme. It is the name of the restriction enzyme produced by bacteria. Why it is very popular? Because it is a very flexible tool, and it has two enzymes. One is this enzyme Cas9, and the other enzyme is what is known as gRNA, guide RNA. This guide RNA is quite a unique tool. Why? Because it guides the it guides the Cas9 where the cut has to be made. It also guides where the joining has to be done. So the accuracy of this CRISPR-Cas9 method is quite high, and because of that it is very, very popular. So CRISPR-Cas9, this tool is used for gene editing nowadays. So CRISPR-Cas9 had come in news when in 2018. So you would have heard that there was a Chinese scientist, He Jiankai. So he produced world's first gene edited babies. So He Jiankai produced world's first gene edited babies, Lulu and Nana. So these babies are resistant to HIV. These babies are resistant to HIV. So he used this CRISPR-Cas9 method to get this type of HIV resistant baby. So here you can see that there is this guide RNA. If you want to, if this is the target, if this is the target DNA, if you want to make a cut, the guide RNA will come, and then guide RNA will guide your Cas9. So Cas9 comes, the Cas9 makes a cut, and it makes joining also. So in this way, this accuracy of this method is quite high. So in 2018, He Jiankai produced this Lulu and Nana. So what he did essentially, that he removed a particular gene from these embryo of these two babies. So let us say this is the DNA. So he removed one gene, the CCR5 gene. So let us say this is the functional part of the gene, and this is your CCR5 gene. So it is said that this gene is responsible for causing HIV. So what he did in the embryo of these two babies, so when they were at the embryo stage itself, he went for gene editing, and he removed these he removed the CCR5 gene, and because of that, so these babies are resistant to HIV, because they are not having this CCR5 gene, so they are not vulnerable to HIV. So that is why from then this CRISPR-Cas9 has become very popular. So So we'll see that where are the other application areas of this CRISPR-Cas9. So nowadays this CRISPR-Cas9 has become very popular, but in order to understand, we also need to understand the different gene editing, the recent gene editing methods that we are using, recent or where we are using this CRISPR-Cas9. So recently you would have heard about cases like in USA a woman was treated of HIV using stem cell therapy. Then we are also trying to develop some cancer therapies using these gene editing tools only. So there is a girl in UK, Alicia. So she's suffering from an autoimmune disease. So what we are trying to do is that we are trying to edit some of the genes in her. So in that sense, it was in news. So there are many items with respect to CRISPR-Cas9 that we can be aware of. So let us look at some of the cases why it was in news. So in one of the case, so let me just take another slide. We'll come to these questions little later. So in this case of Alicia, so Alicia is suffering from an autoimmune disease. So what is happening in her case is that her T cells in our body, they are a part of our white blood cells. Her T cells have become defective. These T cells are also one of the type of T cells are known as killer cells. So what do these killer cells do? They attack the pathogens, they attack the viruses or bacteria, and they kill these pathogens. So these T cells are our defense mechanism. So they are a part of our white blood cells. So what could happen? Why are we calling it as autoimmune? It is because the T cells have become defective, and these T cells are killing the healthy cells. So the T cells have become defective, and defective in the sense they have got mutated. So they have got mutated, and they are killing what? They are killing the healthy cells, killing healthy cells. So what is the cure in this case? So what was done in this case is that this T cells from her body, some of the T cells from her body were extracted. T cells from our body was extracted, and then that we went for base editing. Using CRISPR-Cas9. So when you say base editing, base here means the nitrogen bases, and the base editing was done using CRISPR-Cas9, and the T cells were made healthy. So after base editing, so we got healthy T cells, and now these healthy T cells, they were again inserted back in our body. So what these healthy T cells are doing, these healthy T cells are killing the defective T cells in our body. So what was done in case of Alicia, that we are taking these T cells are the immune cells, or they are part of our immune system, that is specifically the white blood cells. So these T cells are known as killer cells. Their job or their role is to kill the pathogens that come and that invade our body. But sometimes if there are some defects, then these T cells can also kill the healthy cells of our body. So this is happening in case of Alicia. So what was done is that these T cells, some of these T cells were extracted, and base editing using CRISPR-Cas9 was done, and then we got healthy T cells. These healthy T cells were transferred to Alicia, and what we are seeing, these healthy T cells are now killing the defective T cells in Alicia. So in this way, what we are seeing, that her autoimmune disease is retreating. So she is getting cured, but this is not a commercial cure, or it is undergoing trial. So we'll see more such methods in future. So this was the particular case of Alicia, and in USA, let us look at another case where this CRISPR-Cas9 was used. In USA, a woman is suffering from sickle cell anemia. Sickle cell anemia. So what is sickle cell? In sickle cell, what happens that our blood cells, that is our RBC, is circular in shape. So circular in shape, it is called as biconcave shape, or you can just understand they are circular in shape. In sickle cell, the RBC becomes sickle in shape, or half moon shaped, or crescent shaped. So because of that, they clog or they block the blood vessels, so sometimes they can cause very excruciating pain, and also it leads to anemia. Why? Because the amount of the hemoglobin in the RBC drastically reduces, so such people suffer anemia. So there is loss of supply of oxygen to different parts of the body. So this in this case, what was done, that this why people suffer from sickle cell anemia? So there is defective bone marrow. So inside the bone marrow, you have what are known as hematopoietic stem cells. These hematopoietic stem cells lead to formation of your RBC, that is the red blood cell, and the white blood cell. So in such people, what you see, that defective RBC is produced. Because they are producing, the bone marrow is producing defective RBC, we need to produce some or find some other mechanism through which we can produce healthy RBCs. So what was done in her case? So in her case, what was done, that we know, this is the case of Victoria Gray. So in her case, we know that in case of fetus, so when you talk about fetus, when the child is inside the mother's womb, so there the RBC, so the fetus has a different mechanism to produce these hemoglobins or to transfer oxygen. So there we produce what is known as fetal hemoglobin. Fetal hemoglobin. But what happens, that when a child is born, this mechanism switches off, and now it switches to, or it relies on the bone marrow, or on the bone marrow's capacity to produce RBC. So what was done, that we, using CRISPR-Cas9, we are again able to revive this mechanism of fetal hemoglobin. So this was done again using CRISPR-Cas9 only. So you see that CRISPR-Cas9 is being used in many ways, and nowadays we are also trying to develop these cancer therapies. So in cancer therapy, what you are seeing, or in news, so you would be hearing about what is known as immunotherapy. So what is this immunotherapy? So traditionally, or how are we treating cancer nowadays? Either we are using chemotherapy, or we are using radiation therapy. So what is cancer essentially? Cancer means the cells, they start multiplying but they do not stop. So they somehow trick the immune system so that the immune system do not attack these cancer cells. But we are using some chemicals or radiation to stop these cancerous cells. Now we are thinking that we can train our immune system again to kill this cancer cell. So how do we train this immune system? Is by using again this CRISPR-Cas9 only. So we are using CRISPR-Cas9 method to train our immune system. So in cancer therapy, one of the famous cancer therapy that is popular nowadays is your CAR T therapy. CAR T therapy. So in CAR T therapy, what we are trying to do, so in CAR T therapy, what is being done, that this T cells, the Just a second. In CAR T therapy, what we are doing is that we are taking T cells and we are again going for its modification. So we are modifying the T cells. So we are modifying we are extracting the T cells from the patient, from cancer patients. We are modifying the T cells, and these modified T cells can then identify a particular type of cancer. So it will identify the particular type of cancer cells. So and then it will start to kill. It will identify the particular cancer cells and it will kill them. So what we are doing is that we are able to train our immune system itself. So T cells are what? T cells are part of our immune system. So in this we are utilizing this philosophy that our immune system itself has the capacity to fight against these cancers. So this is a revolution in cancer therapy. So we are not relying on chemicals, we are not relying on radiation, but we are relying on our immune system itself. What do these cancer cells do? These cancer cells somehow they trick our T cells or our immune systems so that they do not identify them as a problematic cell. So that is why these T cells, they do not kill the cancer cells. Otherwise, if they are able to identify that the cancer cells are harmful to our body, then they can kill those. But somehow these cancer cells trick the T cells. So this is how we are training it. Another popular method that is going on is what is known as PD1 blockade. PD1 blockade. What is this PD1? So the PD1 blockade, what is the PD1 is a particular kind of protein. This particular kind of protein, this particular kind of protein doesn't allow the T cells to identify the cancer cells. So they are acting like brakes. They are acting like brakes on T cells. Our T cells has the capacity to kill the cancer cells, but these PD1 proteins, they are not allowing the T cells to identify cancer cells. So what we are doing, again, we are using gene editing tools to block this PD1 protein. That is why this treatment is called as PD1 blockade. So what we are doing, that this PD1 was acting like a brake on the immune system. So it was acting like a brake on the immune system. It was not allowing the immune system to get activated or to act on the cancer cells. So what we are doing, we are just releasing this brake by blocking this PD1. So when we are blocking this PD1, now our T cells or our immune system can act against them. So in that way we are able to kill the cancer cells. Again, but in cancer cell, the biggest problem is that these T cells, the modified T cells, cannot kill all type of cancer cells in the body. So if you are modifying it for, let us say, skin cancer, then it can kill for only skin. It can kill only skin cancer cells. It cannot, let us say, kill your pancreatic cancer cells. So if you have to kill the pancreatic cancer cells, you will have to modify it differently. So it will be able to kill only pancreatic cells, pancreatic cancer cells. So that is a limitation. But again, so that limitation is actually can be overcome. Why? Because these gene editing tools are quite flexible. So we can quickly modify them or quickly modify T cells for a particular kind of cancer. So that is the advantage with respect to this immunotherapy. So you should take a note of this immunotherapy. This is a revolutionary field with respect to cancer treatment, and here again we are using this CRISPR-Cas9 only. In other areas of gene editing, so we also need to understand there are other areas. So CRISPR-Cas9 is also used in other areas also, or in general, what are the gene editing tools and what are the application areas. So let us now see some of the application areas of gene editing. So gene editing and its application. Here we are seeing application mainly in health, in let us say human health. So in human health you can identify in two categories. So in what are two categories? So we can go for gene editing in what is known as germ cells, and we can go for gene editing in what is known as somatic cells. So what are germ cells and somatic cells? So germ cells are the cells which are present in the reproductive organs. So they are also called as the sex cells. And what are somatic cells? All the cells in the body other than the germ cells are called as somatic cells or body cells. So gene editing we can go and do with respect to any of these type of cells. So we can go for gene editing of somatic cells, we can go for gene editing of germ cells. So here in this case, let us look at some of the cases like this 3-parent baby problem. So this 3-parent baby issue, what we need to understand that some of the children, they could be born with some of the mitochondrial diseases. So we need to understand the basic concept of this, then we'll look into it. So in this we'll see what is this 3-parent baby and how it is being produced. In this only what we can do, or we can understand cloning also. Cloning, in this we'll try to understand what is this IVF. So if you understand IVF, then we'll have a clear picture of this 3-parent baby. In somatic cell gene editing, we have seen this case of Alicia or Victoria Gray or cancer therapies. So we have seen some cases, the cancer therapies that we have seen. And we'll also look at this stem cell therapy. So there was a news in USA that using stem cell therapy a woman was cured of HIV. So we'll look into this also, but let us just look at this part. So how we are using gene editing in IVF. So you need to understand first IVF, and then we'll see 3-parent baby, that how we are modifying the genes. So what is IVF? IVF means what? In vitro fertilization. In vitro fertilization means that fertilization of the egg and the sperm cell is done in the lab. So because it is done in the lab, so then the next stage after fertilization that is formed is your zygote. That zygote starts to divide and it forms an embryo. That embryo is implanted in mother's womb. But in some cases what you see that there is defective mother's mitochondria. So here we need to understand with respect to 3-parent baby that why do we need to go for 3-parent baby. So why are we going for 3-parent baby? So one of the prominent reasons, or the reason is that what is called as there is defective mitochondrial DNA inheritance disease. DNA inheritance disease. So see, we first need to understand, we said in the initial slide that DNA is present inside nucleus. So we get DNA, or we get genes from both the parents. So we get genes from mother side and we get genes from the father side. So we are getting the nuclear DNA from both the mother side and the father side. Nuclear DNA means the DNA which is inside the nucleus of the cell, but there are some DNA which is present outside the nucleus, that is present only in your mitochondria. So even in your mitochondria there are some DNA present, and this DNA or that is present in mitochondria is passed from mother to child. It is not passed from father to child. So what we see that this inheritance, so mitochondrial DNA inheritance has a particular pattern, that mitochondrial DNA passes only from the mother to the child and it doesn't come from the father side. So what if this mitochondrial DNA is defective in some cases? So what do we do in such cases if it is defective in some cases? So what is the scenario, what is the problem, is that the baby could be born with some severe infections or severe genetic diseases. To avoid that scenario, we go for what is known as three parent baby. So what is this three parent baby? Three parent baby is essentially mitochondrial replacement therapy. So mitochondrial replacement therapy, how, what are we doing here? So here we are going for genetic manipulation only. So we're replacing the mitochondria. So how do we do that? There are two ways of doing this, so let us just see that. So in this diagram you can see, let us say this is the father and this is the mother. So this is the father, this is the mother, and in black what you are seeing, that black here, or full complete body is black. So it means that her in her mitochondria there are defective DNA, or her mitochondria is defective. So you can see that the next generation that is born, so they are having this defective mitochondria. So they are having this defective mitochondria. So it is being passed from the mother to the child. So there are two ways of getting this done, or how do we go for replacement therapy. So in one of the therapies what we do, so we take, so in this what we are doing, let us say this is the mother's egg cell. So in this mother's egg cell, here you can see faulty mitochondrial DNA. This mitochondria is present in the cytoplasm. These mitochondria, some of the mitochondria could be defective, but the nucleus is healthy. So what we have done is that we can remove the nucleus and leave this cytoplasm. So this is nucleus and this is cytoplasm. So we are using enucleation method. So we'll remove the nucleus. So in this diagram you can see that we have removed the nucleus. We have taken another mother. So this is the donor mother. So this is the original mother. She is the donor mother. So we have taken the egg cell of this donor mother. Her what you can see, the egg cell is having all healthy mitochondria or healthy mitochondrial DNA. What we have also done is that her donor mother's nucleus is removed. Why? Because we do not want her nucleus. We do not want this nucleus, we want this nucleus. So we have taken nucleus from this mother, and cytoplasm from this mother. And now what you have got is a new or a reconstructed egg cell, in which you can see that all these mitochondria are healthy. So all these mitochondria are healthy, and you have a healthy nucleus also. So this behaves, or this acts like what, a new egg cell. And now what we'll do, so we'll fertilize this in the lab. So you have the egg cell and you have the healthy mitochondria, and we'll take the sperm cell and we'll fuse, and then we'll get a zygote. So we'll get a zygote. So this is a zygote. So after a zygote is formed, so then after some days, so embryo will be formed. So this embryo will be transplanted into mother's womb. So let us say this is the there was this original mother. So this embryo will be transplanted into mother's womb. So if it is transplanted into somebody else, then that calls for the case of surrogacy. So in surrogacy, the embryo is transplanted not to the initiating mother, or whose egg cells has been given, but to the womb of somebody else. So in this way you can get a three parent baby. So in a three parent baby, you see that there are two mothers and then a father. So reconstructed egg cell, fertilized with sperm in the lab and implanted into the patient. Resulting embryo has three genetic parents. So why we are saying three genetic parents? So from here we have got nuclear DNA, from here we have got the mitochondrial DNA, and from here again in the nuclear DNA, one half of the nuclear DNA is contributed by the father's sperm. So in this way we are able to produce a three parent baby. So this method is also called as maternal spindle transfer method. So this method is also called as maternal spindle transfer method. And what you can see that all the genetic editing or replacement has been done before fertilization. All genetic editing has been done before fertilization. So this is the one way to produce the three parent baby, and there is other way also. So let us look at the another way. So in this case what we are doing that we are taking the mother's egg cell and we are taking the father's sperm cell. So mother's egg cell, so mother's nucleus is here, this is the father nucleus. This stage is called as your pronuclear stage. Pronuclear means just before one or a single nucleus is formed in the zygote. So the nucleus from the mother and nucleus from the father, they just attach side by side. It is a very transient stage, so very short stage, and then it will fuse eventually to form one nucleus. So what we are doing here, we are allowing fertilization first, and there you can see that in the egg cell you have these defective, but we are not interested with the cytoplasm here, we are interested in this nucleus. So what we'll do, we'll extract this pronucleus, we are enucleating this pronucleus, and from a donor mother we are taking a healthy egg cell. We are taking a healthy egg cell. In that healthy egg cell you have all the mitochondrial DNAs that are healthy, that are okay, and then we have removed her nucleus and we have added this nucleus. Now this pronucleus we have added to this cell. So now this will behave like a zygote, and when you give it an electrical shock, then it starts to multiply and it becomes an embryo, and then it can be transplanted to this mother. So this is the second method, and this method is called as your pronuclear transfer method. So sometimes in UPSC they are asking just the scientific name, or they could ask the method also. They could ask the method also, or they could ask the scientific name also. So in this case what we are doing, we are going for fertilization first, and then we are going for your mitochondrial replacement therapies. So these are some of the ways in which we can get the three parent babies. Let us look at some questions. So let us just try to solve this question. In the context of recent advances in human reproductive technology, pronuclear transfer is used too. So let us just try to solve this question. I hope pronuclear transfer is used. Yes, you can go ahead and so in the context of recent advances in human reproductive technology, pronuclear transfer is used for, so it is used for prevention of mitochondrial disease in offspring. So this is the correct answer. So Let me just bring some more questions. Let us look at some of the questions, then we'll proceed ahead. Let us look at this question. A recombinant DNA technology. Recombinant DNA technology, genetic engineering allows genes to be transferred across different species of plants, from animals to plants, from microorganisms to higher organisms. Select the correct solution using the codes given below. So 1 only, 2 and 3 only, 1 and 3 only, 1, 2, 3. So you can give it a try. Yes, sir. Okay, so let us look at the answer here. So recombinant DNA technology. So again, what we are saying, that we are talking about genetic engineering. So it says that allows the genes to be transferred. So across different species of plants. So yes, we said that the principle of biotechnology is what, that because we have evolved from common ancestors, so it means that biologically and chemically we are very similar. So all organisms are essentially using your DNA as method to code information for one generation to another generation. So we can transfer genes or from one species of plants to different species of plants. It is not only about plants, but we can transfer it from plants to humans to animals to any other organisms. So this is a correct statement. From animals to plants, that is also a correct statement. From microorganisms to higher organisms, yes, we can do that. We can transfer some genes from bacteria to higher organisms. So this is also true. So the correct answer in this case is your D, that is 1, 2, 3. Let us look at some other questions. Let us try to solve this question. With reference to recent developments regarding recombinant vector vaccines, consider the following statements. Genetic engineering is applied in the development of these vaccines. Bacteria and viruses are used as vector. Okay, so yes, you can try. With reference to recent developments regarding recombinant vector vaccines, consider the following statements. Genetic engineering is applied in the development of these vaccines. So yes, it is a very generic term. So yes, genetic engineering is applied in the development of these vaccines. Bacteria and viruses are used as vectors. So we have seen that bacteria can be a vector, virus can be a vector, bacteriophages can be vector. So there are many such organisms that can be used as vector. Fungi can be used as a vector. Your yeast can be used as a vector. So there are many such organisms that can be used as a vector. So this question had come during the COVID time. So we have what are known as your viral or virus vectored vaccine. The virus vectored vaccine, the most famous virus vectored vaccine is your Covishield. So with reference to that, most of us, they think that vectored vaccines are only virus vectored vaccine. No, the vector can be anything. It can be a bacteria, it can be a virus also. So we need to understand that vectors can be anything. Vectors are just what medium to transfer the desired genes from one organism to another organism, or from a particular organism to the host. So the correct answer in this case is both 1 and 2, that bacteria and viruses are also, you can be used as vectors. Let us look at this question. So this is an easy question. Let us try this question. What is Cas9 protein that is often mentioned in the news? A molecular scissors used in targeted gene editing, biosensor used in the accurate detection of pathogens in patients, a gene that makes plants pest resistant, a herbicidal substance synthesized in general modified crops. So we have seen that Cas9 are molecular scissors, so they are helping us in targeted gene editing. So it is one of the most prominent tools that we're using nowadays for gene editing, that is the CRISPR-Cas9. So the correct answer in this case is your A, that it is a molecular that it is a molecular scissor. It is a molecular scissor used in targeted gene editing. Okay, so let us look at other questions. Now try this question again. This question you can answer just by relying on what we studied, that what is the principle of biotechnology. Principle of biotechnology. Yes, you can try. So in this question, what it is saying, consider the following statements. Genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. A person's genome can be edited before birth at the early embryonic stage, and human-induced pluripotent stem cells can be injected into the embryo of a pig. So let us look at each statement and let us try to find the answer to this. So genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parents. So yes, so genetic changes, so genetic tool or gene editing tools are very powerful tools, and they can be used to modify your eggs, your sperms before fertilization, after fertilization, or during the embryo stage, or after it has been implanted into the uterus also. So in all those scenarios we have this potential to change the genes. So yes, it is true that genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. So this first statement is correct. Now let us look at the second statement. A person's genome can be edited before birth at the early embryonic stage. So yes, so it can be edited at early embryonic stage also. So if you remember the case of He Jiankai, so he produced gene edited babies or HIV resistant babies using he edited the at the embryo stage only. So it is possible to edit at the embryo stage. So this is also correct. This is also correct. Human induced pluripotent stem cells can be injected into the embryo of a pig. So UPSC does this, that they will throw you some bombastic concepts or terms and they will put you off guard. So what you have to do, you have to be very patient in such type of statements. So this human induced pluripotent stem cells can be injected into the embryo of a pig. So timrantak timrantak is saying that 3 is got to be wrong. I think 1 only. 1 only. Why do you think the second statement is wrong? So we studied and we said that even at the embryo stage, gene can be edited. See, gene editing is a very powerful tool. There is no restriction or limitation with respect to when you can do it. So theoretically, we have no restriction. Practically, we might not do it at some stages, but theoretically, we can do it at any stage. At embryonic stage, we have already done. So there are many cases where we have done changes at the embryonic stage. So He Jiankai, the Chinese scientist which produced the first gene edited babies, he did modification at the embryonic stage only. So at the embryonic stage, he removed a particular gene that makes a person vulnerable to HIV. So he removed the CCR5 gene at the embryonic stage only. So second statement is also correct. Are you able to get this, Timrantak? So second statement is also correct. What about the third statement? Third statement, that human induced pluripotent stem cells can be injected into the embryo of a pig. Is this statement correct or incorrect? Let us say we haven't discussed about stem cells, but still you can answer this question. Why? Because see, when you're talking about human induced pluripotent stem cells, it is a kind of a cell only. It is a kind of human cell only. So what this statement is saying, that can human cells be injected into pigs? Can human cells be injected into pigs? That is the question. So what do you think? Can human cells be injected into pig? Recently in USA, there was a case of what is known as xenotransplant. Xenotransplant. So in xenotransplant, you can transplant some parts of some organisms into another organism. So in USA, what was done, that the heart of a pig was transplanted into a human in USA, but after 2 months he died. But theoretically it can be transferred. Practically it was done, but there was immune response, so the person died. So your pig is also containing what, four chambered heart. But why here we are looking at more fundamental step, that can human cells be injected into embryo of a pig? So yes. Why? Because biologically and chemically we all are similar. Biologically and chemically we all are using DNA only as a means to transfer genes, or as a means to transfer trait from one generation to another, as a means to produce proteins. So we are using the same method. That is why it is possible to transfer human cells into pig or pig cells into humans. So even this statement is correct. So in this case, the correct answer is your D. That is, all the statements are correct. Okay, so let us look at another question. These questions we will deal little later, or let us just try to do this also. In the context of the developments in bioinformatics, the term transcriptome, sometimes seen in the news, refers to sometimes seen in the news refers to. So all of you can go ahead and try this question. What do you mean by transcriptome? Yes, what do you think? What is the answer here? Okay, so if you study protein synthesis, see, protein synthesis is quite important for us to understand. So in protein synthesis, what is happening? So our every cell has capacity to make proteins. So inside cell, if you see, then there are ribosomes. Those ribosomes are called as your protein factories. So ribosomes make protein, but when they will make protein, when they will get information from the nucleus that which type of protein has to be made. So how does it gets the information that which type of protein has to be made? So there is something called as messenger RNA. The role of the messenger RNA is to tell the protein factories, that is the ribosomes, to make which type of protein. So your the nucleus, the DNA opens up. Inside the nucleus you will see DNA. Those DNA have information regarding which type of protein has to be made. So your nucleus then makes from DNA some messenger RNA. So from DNA you get some messenger RNA inside nucleus. And this step is called as transcription. This step is called as transcription. So in this transcription, what is happening? From DNA you are getting mRNA. This mRNA will go to ribosomes, and then the ribosomes now have the information of which type of protein has to be made. So then we'll start the protein synthesis. So your DNA, or inside the nucleus, our body, or this nucleus contains all the information regarding all the types of proteins that our body has to make. So this mRNA that you get is essentially called as one transcript. So it has one transcript regarding one type of protein. So our body needs different types of protein, so we need different transcripts. So what is this transcriptome? Transcriptome means a collection of all the transcripts that is possible. So it refers. So what is the answer in this case? It is not this one. A range of enzymes used in genome editing. It is the full range of mRNA molecules expressed by an organism. So this is your correct answer. The description of the mechanism of gene expression, this is not correct. A mechanism of genetic mutations taking place in the cell. So protein synthesis we should understand. So it is this process that is the first process is transcription. So transcription is the first process, and the another process that is involved is your what is called as translation. So what is happening in translation? So these mRNA is giving information to ribosomes for which type of protein has to be made. These ribosomes then actually make those proteins, and this process is called as your translation. So this mRNA is quite important from exam point of view. Why? Because we have developed mRNA based vaccines. So what are these mRNA based vaccines? Do you know the examples of the mRNA based vaccines? So one of the example is your Moderna vaccine and Pfizer's vaccines. So they were the first one who made vaccines for COVID, and they were using this mRNA based vaccines. So what is this mRNA based vaccine? We need to understand this also, but let us just try to answer a related question only. So let us try to answer this question. RNA interference technology has gained popularity in the last few years. Why? RNA interference technology has gained popularity in last few years. Why? Yes, anybody? Okay, so this RNA interference technology has gained popularity. So, what is this RNA interference? So here you see that your DNA, from a DNA you are getting mRNA. This mRNA, this process is happening inside nucleus. So this is happening inside nucleus. Inside nucleus, then this mRNA moves outside. It is moving outside to reach the ribosomes, and then translation occurs. What we are saying RNA interference is that this mRNA is made, but some agent come and interfere with the mRNA. Because they are coming and interfering with the mRNA, the mRNA is not able to pass information to the ribosomes to make which type of protein. So there is some interference with the mRNA. Because there is some interference with the mRNA, the proteins is not formed. So that essentially what we are saying is that genes are not able to express itself. Genes, how do genes express itself? Through formation of proteins only. So when they form protein, they will express themselves, but if they are not able to form proteins, they are not able to express themselves. So genes are there, and the genes led to formation of mRNA also, but somehow some agents interfered with mRNA, and it is not leading to formation of proteins. So genes is present there, but protein is not being formed. So this is what is happening, is that the gene is essentially, a gene is being silenced. So this is known as gene silencing. This is known as gene silencing. So what is happening? I'll get to the question. It is used in gene silencing therapies. Yes, it is used in gene silencing therapies. So it can be used in developing therapies for treatment of cancer. Yes, it can be used for developing treatment for cancer. Why? Because in cancer, the cancer cells, they start multiplying, but they do not stop. So if we can silence some of the genes, then we can stop the cancer cells also. So it can be used to develop hormone replacement therapy. Hormone replacement therapy, actually we need to add hormones. We need to add hormones to replace. So this therapy can switch off something, but it cannot switch on something. So this is not true. It can be used to produce crop plants that are resistant to virtual pathogens. This is not virtual, this is viral pathogens, or viral pathogens. So it can be used to produce crop plants that are resistant to viral pathogens. So yes, so there could be presence of some genes in the plant that is making it vulnerable to pathogens. So what we can do in such scenarios is that we can silence those genes. So in that scenario, yes, it can be used for this. So this is correct, this is correct, and this is also correct. So these are some of the questions that we have seen. Okay, so now let us look at this cloning also. So what do we do in cloning? So let us look at this cloning. So what are we doing in cloning here? So we have seen the 3-parent baby case, and we have tried to understand that how we can remove the mitochondrial DNA defect from mother to her offsprings. Now we'll see what is cloning. So in cloning, what we are trying to do is to make exact copy of a particular organism. So here what we are seeing in this slide is that we want to make exact copy of, let us say, this pig. So we want to make a clone of this pig. So how the clone is made? So what we do is that we will take the skin cell or any somatic cell from the adult. So we have taken the skin cell from the adult, and what we have done is that we have extracted from this skin cell, we will extract the nucleus. So from the skin cell we'll extract the nucleus. And here we have taken an adult female, and from the adult female what we have done is that we have taken an unfertilized egg cell and we are removing the nucleus. So this nucleus is removed. So this nucleus is removed. We are only taking the cytoplasm of this egg cell, and now we are fusing the egg cell with the skin cells nucleus, and what we are getting here is that cell divides and we start getting an embryo. So what we have done is that we have not taken a sperm cell, we have just taken a nucleus from organism of which we want to make a clone. So we have taken the nucleus from the skin cells or from any somatic cell, and we have fused it with cytoplasm or cytoplasm of the egg cell of a mother, of a female. So after that what we see that it behaves like a new zygote. So this nucleus and this cytoplasm, it behaves like a new zygote and starts to divide to become what is known as embryo. Then this embryo is transplanted into an surrogate mother, into the womb of a mother, and after the gestation period what we see that we get a cloned animal. So this is how the cloned animals are made. So the first cloned animal that was made was Dolly the sheep. And do you know that in India so there are some doubts. Let us take those doubts. Aditi is asking, so what can be other somatic cells? Somatic cells are your body cells. You just understand that the cells inside are reproductive organs. They are considered as the germ cells or the sex cells. Any other cell, they are somatic cells only. Your skin cell, your blood cell, your hair cell, your bone cell, your heart cell, your liver cell, any cell that you think of, they are mainly your somatic only. Only inside the reproductive organ, the cells that we have, they are called as germ cells or sex cells. Is that clear, Aditi? Okay, so Timrantak is saying all cells except ova and sperm, somatic cells, I guess. Not sure. Yes, see, ova and sperm, they are formed from your sex cells. They are also one form of your sex cells only, but they are being formed from the sex cells. Your sex cells will have 46 chromosomes, but your ovum and your sperm cells do not have 46 chromosomes. They have half the number of chromosomes. So your egg cell has 23 chromosomes, your sperm cell has 23 chromosomes. So when they fuse together, then again in the zygote you have 46 chromosomes. So we are made from, or we have half genetic characteristics from our father side, half from mother side. So we have 46 chromosomes inside our nucleus. So 23 comes from the father side, 23 comes from the mother side. So your egg cell and your sperm cells, they contain half number of chromosomes, that is 23 chromosomes in each. But these sex cells, they have 46 chromosomes, but during the formation of gamete, that is during the formation of the egg cell and the sperm cell, it undergoes a particular kind of cell division and the number of chromosomes reduces to half. Do you know what is that cell division process called as during formation of gametes? When you say gametes, essentially it means formation of the egg cell and the sperm cell. So that process is called as gamete formation, and that is formed using meiosis. So that is formed using meiosis. Okay, so let us just try to understand this cloning once again. So is the cloning part clear to all of you? So what we are doing is that we are taking the skin cell or the somatic cell. In this case, we are not taking a sperm cell. In this case, we are taking an egg cell, but we are not taking a sperm cell. We are taking a skin cell. From that we are extracting the nucleus. Our every body in our cell, they contain the same genetic information. So they all have 46 chromosomes. So because they are having 46 chromosomes, when we want to make a clone, we are just taking some of the somatic cell. We have those 46 chromosomes, and then we are transferring it to a egg cell. So we have removed this egg, removed the nucleus from this. We have fused the nucleus, and then we have got the embryo, and we have transplanted the embryo into the surrogate mother, and after the gestation period, this cloned animal is formed. In India, which was the first cloned animal, first live cloned animal, so there was this Garima, the sheep, that was the live cloned, the first alive cloned animal that was formed. So in this, what type of questions can you expect? So in this, the type of questions that are asked is basically the scientific names sometimes can be asked. So cloning is also known as somatic cell nuclear transfer technology. Why it is called as somatic cell nuclear transfer technology? Because we are taking the somatic cells of the organism which we want to clone. So we are using the somatic cells to make clones. So this is also known as somatic cell nuclear transfer technology. So what is the answer in this case? It is for reproductive cloning of animals. So this is the answer in this case, for reproductive cloning of animals. Okay, any other doubts? If you have, you can ask, so we can devote some time for doubt clearing also, or we'll switch to stem cells, stem cell therapy. So C, yes, C is the correct answer. Reproductive cloning of animals. What do you think will be the, let us say in this question or in this scenario, what do you think will be the gender of this? Gender of this? Will it be different from this, or will it be same as the skin from the animal that we took the skin cells? What will be the gender of this pig that you have got here? So because it is a clone, so its gender will also be same as that of the initiating organism. So because we are taking the nuclear DNA has 46 chromosomes. The last two chromosomes, that is your 45th and 46th chromosome, are known as the sex chromosome. So let us say if it is XX, then it is what? It is female, and if it is XY, it is male. So we are talking about female and male. So in this 46 chromosome, so you have this information regarding gender also. Yes, so the gender of this will be same as that of this organism. Why? Because we are making a clone, so here the gender related information is also passed. So the same, it will be of same gender, it will be of same gender. Okay, are we going for human cloning? Are we going for human cloning? Are we going for human cloning? Yes, no? Okay, so let us see stem cells then in brief. and we'll discuss more tomorrow. But let us just understand what are stem cells. What are stem cells? What are stem cells? So what we are seeing here in the slide, that we are saying that they are body's master cells. Body's master cell. What are the concerns with respect to, let us say, human cloning? So human cloning, there are many concerns. First of all, biologically, the concern is that the success rate of cloning is very low. So in most of the cases, you see that the failure rate is very high. So that is not advisable, or that is not a good way to proceed. Secondly, what we'll see, that there are many ethical concerns. So human cloning, most of the religions, they are opposed to the idea of human cloning. Why? Because then we will alter the course of nature. We can alter the course of nature, so we would become like God. So major religions across the globe, they are not with the idea of cloning, so they are against the idea of cloning. And there are some other concerns, like already we are 7 billion plus, so if we start going for cloning, so there will be more crunch on resources. And if you look at these type of methods, so these methods, who will have access to such type of methods? So only rich people would be having access to such type of method. So this could be problematic. Only developed countries could be using it. So in developing countries, you won't be seeing cloning. Even in developing countries, those who are rich, they could have access to this type of technology. So it can increase the divide between the rich and the poor. That is a concern. And there could also be, in future, if you allow for human cloning, there could be gender specific cloning also. So in India, you see the incidences of female feticide. So if you allow for cloning, so there could be clones of just males also. So this is a concern with respect to this. Cloning may turn out to be a disaster. Yes, Aditi, that is true. It could turn out to be a disaster, because you're trying to manipulate genes, and in that way you're trying to manipulate evolution itself. So it can be disastrous also. So that is also one of the reasons that we are not allowing for human cloning. With respect to personalities also, it is commented that if we go for cloning, so because the genes will be very similar, can we get humans which will have different personality if they are having exactly same genes? So in many studies that we have seen, that if the genes are very similar, or what we have seen in case of identical twins also, that because the genes are very similar, somehow the personalities also are very similar. So we do not want that also, that the personalities should be same. That is from the personality perspective. Even from biology perspective, the biggest concern is that when we start going for cloning, so we are reducing the genetic diversity of the population. So we are increasing what is known as inbreeding depression. So this could make the genetic pool very vulnerable to future attack by a virus or a fungi. So if a virus like COVID comes, it can wipe out all the clones at one go. So genetic diversity, more the genetic diversity, so the richer gene pool we have, more fitter our population is. So if we go for cloning, we'll be using one gene pool only, or one type of genes only. That is also a threat. So that is also a concern. Legal difficulties, such as using yes, so there would be legal difficulties also. So if genetically similar, so in cases of crimes, or in cases of paternity issues, how will we be able to resolve such issues? So there will be genetic contamination also. Yes, so these are some concerns. Okay, so what do you know about stem cells? What do you know about stem cells? So why these stem cells were in news? So this was in news because in USA a woman was cured of HIV using stem cell therapy. So what was done is that stem cells from a donor person who was resistant to HIV was inserted into this particular woman, and what we are seeing that HIV in her body is retreating. So what is this stem cell? Stem cells are known as body's master cell. Why they are called as body's master cell? Because they can become any other type of cell. So when we are single, when we are formed, so when we are formed, we are just a zygote, one cell only. After that it starts to divide to form multicellular, or we form an embryo. But we are not just multicellular, or we multicellular, and we are not only one type of multicellular. So our body has different types of cells in our body. So we have your blood cell, you have the heart cell, you have the nerve cell, you have the liver cells. In liver cells also you have so many cells. So we have multicellular liver cells. So how from a single zygote our body is able to make so many different types of cells in the body. So the idea here is that the embryo, they have what is known as stem cells. These stem cells in this diagram you can see, they can differentiate to form liver cells. So they are like your body's master cells. So some of the stem cells will become liver cells, some of the stem cells will become your blood cells. So this what you're saying is RBC, this is your WBC. Some of them can become your cardiac cells. Cardiac cells means the heart cells. Some of them can become nerve cells, some of them can become bone cells, some of them can become your brain cells, or any other type of cell that you can imagine. So these stem cells, they are known as what? They have the capacity to become different type of cells. So we have different types of stem cells. So we further categorize stem cells into different types. So we have what is known as embryonic stem cells. What is this embryonic stem cell? Embryonic stem cells are the stem cells which are found in the embryo. They are also called as pluripotent. So in the previous diagram, what you saw, that those stem cells has the capacity to become any other type of cell. That is why it is called as embryonic stem cell. That is why it is present in our embryo. So inside an embryo you will find these pluripotent stem cells, though they can become different type of cells in the body. Then we have what is known as adult stem cells. These stem cells, they are not pluripotent, they cannot become any other type of cell, but they have a limited capacity to become that type of cell only. So our liver, let us say if you take the example of liver, so inside liver also you will find stem cells but these stem cells can become liver cells only. So they are known as adult stem cells. Then then we have this induced pluri Yes, Timrantak is asking about, so what is the difference between pluripotent and totipotent stem cells? See, these totipotent stem cells, they can form more than 80 types of cells. They are the most powerful stem cells. They can differentiate into more than 80 types of stem cells. This is about totipotent. Pluripotent, they can also become different types of cells in our body, but they do not have capacity as totipotent. Totipotent is just formed after your zygote has been formed, and then you get a stage which is the blastocyst state. So it is in the very initial state only you have these totipotent stem cells. Then when the embryo is formed, then inside the embryo you find the pluripotent stem cells. Is that okay? So if you have any other doubts, so we'll devote some time to doubts, and we'll continue this biotechnology tomorrow. So we'll try to see the application of biotechnology in agriculture sector also, and in some other health sector also, that we'll see how vaccines are being made. So we'll try to understand the different types of vaccines that we are making. Okay, so what we are saying, that there are embryonic stem cells, they are found inside the embryo. Then adult stem cells, they are found inside the adult organs. So if your liver has been formed now, so there are some stem cells inside liver also. So there are stem cells inside heart also, there are stem cells inside bone, blood. So everywhere you will find stem cells. But with aging, what we see, that the number and quantity of these adult stem cells starts to reduce greatly. That is why those damages, the damages to the organs, they are not cured easily. It is also one of the reasons said that since the amount or the number of the adult stem cells start to decrease, that is why we age, and that is why we die also. So that is an area of research. Now this what is this induced pluripotent stem cell? What is this induced pluripotent stem cell? So what we are doing is that in labs we want to convert adult stem cells into pluripotent stem cells. So we want to create some conditions. Under those conditions, adult stem cells, they convert into pluripotent stem cells. Then we can convert into different cells, different cells in our body. So it is induced because it is done in the lab. That is why we are calling it as induced pluripotent stem cells. Okay, so we'll see more regarding stem cells tomorrow, that where do we get these stem cells, and what was done in this case of this woman who was cured of HIV. Okay, any doubts for today? So we'll stop the session for today.